Welcome once again. And now we'll have Aparajita Lahree. She entered Soapbox as a PhD student in Maria Mota Lab in Instituto de Medicina Molecular, and she worked in molecular parasitology, but she recently defended her PhD thesis. So congrats, Aparajita, and the floor is yours.
Thank you so much for the kind introduction. I'm going to start sharing my screen. Please let me know if you see everything properly.
All good, yeah.
Right, so hello everyone, I'm Aparajita, and yes, I recently defended my thesis. I'm here to speak to you today about the theory of almost everything, mostly regarding what is it that encourages us as scientists and the not so nice things as well in science. So let's start. First of all, who am I and what exactly do I do? So, as Martha kindly introduced, I am trained as a molecular parasitologist, which means I study parasites that infect human beings on a molecular level to understand how they function in order to know how to kill them. My specialization is in the area of malaria, which is a disease caused by a protozoan called Plasmodium. Now, you may wonder, okay, we've heard of this disease before, but what is it and really why we should study it? So malaria is a disease which affects more than half of this world, and it's the most ancient human disease. And till date, there is no standard vaccine for everyone. The World Health Organization in 2019 indicated that 400,000 people lost their lives to this disease, most of whom were children less than 5 years of age. So, of course, it is of our concern to investigate this, and let us start that by understanding how this disease really works. So in humans, the disease is started upon the bite of an infected mosquito that delivers these parasites into the human when taking a blood meal. As you see here as green spindle-like structures, these parasites enter the blood circulation and are carried to the liver. They really like the liver. In the liver, they move through several liver cells called hepatocytes and eventually find one that they really like and reside inside a special compartment called the parasitophorus vacuole or PV. It is hiding in this environment that masks it from the host or the human's immune system. In the meantime, the parasite divides multiple thousand times to give rise to thousands of daughter cells, which will eventually be released into the blood. In blood, these daughter cells are capable of infecting red blood cells in a cyclic fashion, giving rise to the pathology associated with malaria, which is fever, chills, can lead to organ damage, or if untreated, even brain damage and death. In this cyclic form of the disease, some forms do give rise to gametes, which when taken up by the mosquito, an uninfected one, leads to the completion of the life cycle of the parasite, thereby completing its life between two hosts or one host and a vector, and ready to infect the next host. So with this, let us try to understand how is it that we study this system. No, we don't drag humans to our lab, nor do we have like fancy mosquitoes. We do, however, have the technology in today's day and time to grow human cells outside of the human body on a petri dish in special medium called culture medium. It is also in this medium that we add synthetically the parasites that we isolate from the mosquitoes and allow for the infection to happen all in vitro or outside of the human body on a dish. Once the parasite has done its job and entered its favorite cell, we add fresh medium and we really check out what this parasite is doing. Indicated in this cartoon as an orange blob, this parasite is initially really tiny and eventually starts stealing resources from the hosts and undergoes cell division to grow into a bigger and a big parasite which completely destroys the structure of the human hepatocyte itself, making it a complete zombie full of the daughter parasites ready to break out. This process can last a few hours to a few days. Now, of course, neither the cells that we work with nor the parasite are visible to naked eye. Hence, we must use specialized machines to do this. And the one that I most frequently used are called microscopes. You must have also used them in your high school to look at roots and leaves. But this one, however, is called a confocal fluorescence microscope and has slightly advanced optics that allows us to see light coming out of the parasite or the host cell, whichever has been labeled for it, and take multiple photos fast in time so that we can really look at life happening. Let me show you a quick video short here where you can see in magenta a host cell on a dish infected with a parasite marked here. Using these machines, we are able to very quickly monitor thousands of such videos that allows us to investigate exactly how the parasite engages with its host and what we must do to disturb the parasite's development. I will now show you the infection in a real liver tissue, where you can see in green the multiple liver cells packed tightly together and their nucleus marked in red. The white boxes will show you the parasite, and take a look at how big they grow in a matter of hours to a few days, completely hiding from the host immune system and giving rise to a disease which is highly fatal. Let's zoom into one of these parasites. As you can appreciate here, the parasite is the big green blob, full of tiny little thousands of daughter cells, which would eventually give rise to the blood infection and can be extremely fatal. Okay, so we use fancy techniques to study this parasite, and so on and so forth. What is the outcome of all of this? Well, the outcome is the drugs, vaccines, and therapeutics that you come across in hospitals or at market. And these all have their roots somewhere in a basic science lab or a basic physics or chemistry lab, where a scientist made an observation through repeatedly monitoring a certain system and helped identify what to alter in order to affect a certain event. And in our case, that is, we want to really stop the parasite from developing so we can save the human. You must have recently heard that a malaria vaccine is being developed in Portugal by the lab of Miguel Prudencio in Instituto de Medicina Molecular. And this is the result of decades of research and looking at all these host-parasite interactions. Then that brings me to a question. Have you taken a vaccine recently? I think I took two. Maybe we can do a small poll here to see if any of you have taken a vaccine. Maybe you want to give me a quick answer. Wow, superb. If you're interested to learn a little more about the vaccines, do join me in the meeting room later. Let's proceed now. So what really makes a scientist? Now, it all starts with a lot of curiosity, followed by a lot of motivation, both of which should help you develop a lot of perseverance to keep trying after failing repeatedly. And let me tell you, scientists fail 99% of the times. You also must be very willing to learn, and as well as be part of an ecosystem. So what is this ecosystem? Well, in high school, it starts with selecting in your 11th and 12th standard science as a specialization. And 2 years later, we then move into our first degree, a licenciatura or a bachelor's degree, where we learn the theoretical basics of biological, physical, chemical sciences or engineering. After that, which can last for 3 to 4 years, we move into a specialization or a master's degree where we are exposed to cutting-edge technology in the field and taught how to make and interpret results. This can then be followed by the most specialized degree, which is a PhD or a doctoral degree, in order to use everything that we have learned so far to make our own novel discovery, which can take anywhere between 4 years to 7 years before you become a professional scientist. So how does science relate to people? Well, science is made by the people for the people, and we cannot exclude the fact that it also has problems that people do. For a very long time, science was looked at with a lot of doubtful eyes, and there was historically complications regarding racism, sexism, and some form of elitism that did not give everybody equal opportunities to pursue this as a career, nor did it give equal opportunities for the dissemination of these discoveries into other people or so-called developing countries. Elitism was also a problem where scientists form a bubble of so-called, intellectual superiority, and they get cut off from the very people for whom they are developing this science or this whatever technology. So how can we then develop high impact science? How can science be meaningful if such is the environment? So thank God, in today's day and time, we have several platforms, including Soapbox Science, which are doing a wonderful job in spreading awareness about diversity, inclusion, about tolerance and accessibility. If you want to talk to me more about this, please join me in the meeting room, and thank you so very much for your attention.
Thank you so much, Aparajita, not only for talking about your work, but also for bringing up these issues that are still present in science. So now we have some time for questions, I think. I want to remind the participants that you can do the questions in the Q&A section or by raising your hand here in the raise hand option in the lower panel of the screen. So Denise, if you want to start.
Thank you. So yes, we have one question from Patricia Fraga. She's asking for permission to talk. I think you already have it. You just need to unmute yourself.
Hi, hello, can you hear me?
Yes, I can hear you. Hello.
Hi, Aparajita. Thank you so much for your presentation. It was really clear. It was actually really enlightening about the theme. So I have two questions, being that the first one is the parasite an actual cell, or is it a virus or something like that? And the second one is that, well, you specified that people in high school use microscopes. So I also work a lot with children, and usually they ask, what type of safety measures do you need to work with such a dangerous parasite?
Thank you so much for your questions, Patricia. They're really great. For the first one, the parasite is indeed a cell. It's a eukaryotic cell, and it's a protozoan, which means it's much behind us in the evolutionary tree, but it is a cell itself. For the second question regarding protection, absolutely, we are working with something that causes diseases in humans. Some of the measures that we take is most of the studies are done in a parasite that cannot completely infect a human. So they are mostly optimized for rodent systems. So even if we do have an exposure, there's no way we would get sick with it. The second is we have to follow certain safety guidelines, which every scientific institution has. You are not allowed to take a living parasite without proper safety restrictions. You must have heard about biosafety levels. So all equipments are equipped or are provided with that, and we have to take care about spillage and exposure. So we are completely safe. Of course, on top of that, we always have our protective gear, but that is absolutely like completely taken care of in these research institutions. I hope I answered you.
Yes, it was really clear. Thank you so much.
Thank you so much.
Deixem-me só fazer aqui um pequeno comentário para os nossos participantes em língua portuguesa, que é, se por algum motivo quiserem fazer alguma pergunta à Aparajita e não estejam confortáveis para fazê-lo em inglês, mandem-nos a questão que nós traduzimos, não há problema. Basically, Aparajita, I was just saying to our Portuguese listeners that they can do the questions in Portuguese and we will translate if needed. Thank you.
Thank you.
Thank you. We still have time for some questions. I don't see anything in the Q&A, but I have a curiosity. 5 years ago, more or less, I was talking with one of the people that was also working in the vaccines for malaria, and they were saying that the effectiveness, I think, is how I say, it was like 50% and not like more than that, or less than 50%.
The current vaccine?
Yep.
So the present vaccine?
No, I don't know if they already did some improvement, but it was, I think, 48 or something %.
Till date, there is no single vaccine that can be used by every age group. So the one you have is only for babies less than 3 years of age. With the maximum efficacy around 40% is what they could get, but it's not lifelong protection. So that is a problem. And therefore, there's a huge surge to look for vaccines which can be given to everybody and which can preferably provide lifetime protection.
Okay, I see. So we have also one question from one of the participants. He's saying, or she's saying, what do you think about the malaria vaccine recently approved by the WHO?
So malaria is a very sensitive issue. And yes, even when the actual biological data is not super strong, the WHO does encourage it. So the current one that has been approved does show better performance than the one that's already in the market, that is marketed by GSK. However, we cannot make any full comments because the trials are not completely done. the vaccine is not just age-based classification, it's also dependent on the race, because different people have different genetics and it can work differently. And they also need to do the safety trials, which means I give you vaccine once, you're fine, but if I give you 3 times, you get sick. We cannot use such a vaccine. At least if we have the time to develop something better, they will. So at this moment, I think I will not make a lot of comments on the current vaccine that has been approved by the WHO. We will wait in time and we will see.